Hello, I'm Dr. Daniel Cameron, and have the pleasure of working with an old colleague. And she just had an article published by the group she's working with. It's Holly Ahern. I've known her for years, and she's right now a Chief Scientific Officer for ACES. And we're going to be talking about a newly published paper about something really dear to me, to my practice, on how to come up with a better blood test so I don't have to rely only on clinical judgment, only on the Western blot, and only on the ELISA. So let me have Holly Ahern introduce you. Thank you for coming and joining me.
Oh, I'm very happy to be here. Yeah, we go back quite a few years, 15 or so years back when my daughter was first ill, and I met you and I met all the other Lyme doctors in New York State because we worked on legislation to try to get to change the landscape for medical practice in New York State. So that was a long time ago. Lot of things have happened since then.
Oh, I was young once. That's it. It's
Right, me
so that's
too.
why it's great is that you have all these hats, including you worked in microbiology for some time, and you took
Yeah,
those skills
I'm
over to
still
where you
doing
are
that. I'm
now.
still a professor of microbiology. Yep.
Yeah, and so you've had plenty of times to teach, but it sounds like you transitioned or pivoted over to a firm that's working on a test, the long sought after test. So I thought I'd get to know test better by exploring with you what you've been up to. So why don't you tell me a little bit about how it came about that you moved from being a professor to this firm you're working with?
Well, so for the record, I'm a co-founder of Aces Diagnostics, and it came about because about maybe 12 or 13 years ago, I met up with a person who was actually in Arizona, living in Arizona, but the same story as mine with her daughter being very, very ill. And she was actually, because Arizona, they really were not well versed at all with Lyme disease. So she ended up flying her daughter from Arizona to New York State to see a physician in New York State. And when that happened, the physician that she saw actually introduced us because she had a foundation, a charitable foundation of 501(c)(3), and she wanted to change it over to do Lyme disease research. And so, she asked if this doctor knew any microbiologists, and because I had been working with all of you for so long, he gave her my name, and then, the rest is sort of history. So we joined forces. The foundation that she founded was Focus on Lyme Foundation in Arizona, and for since that time, we've been raising money and funding research, small scale research projects, just to kind of see if there were any technologies out there that could be, applicable to the Lyme disease situation. Because as you know, the state of Lyme disease diagnostics is very bad, very poor. And I mean, one of the reasons why our daughters didn't get diagnosed and ended up with a chronic form of the illness was because the testing was not where it should have been. So as a result of the research that we performed, our research studies, and I have to say we tried everything. We tried RNA sequencing. We went from there to looking at microRNA sequencing. We tried whole genome sequencing. We tried culture. And by we, I mean, we funded these small scale studies with samples. We started our own biorepository. In New York, obviously, we use New York State for that. And because we had the blood samples and we had, people who were willing to work with us. And as a result of all that, we came up with a couple of things, but then, we crossed paths with Dr. Monica Embers at Tulane University, who has been a leader in Lyme disease research for a long time. And what she was able to show, she had already developed we call it the 5 antigen panel, right? So in her research with non-human primates, she looked at their immune response and found that there were 5 antigens that did a better job than the existing diagnostic testing. And we had done a scale study on something called an immunosignature, which I could spend all day talking about that, but I won't. But it's agnostic profiling of the immune response, right? So in other words, not looking for one specific antigen, but or antibody, just looking to see what happens in a person who has Lyme disease, what kinds of antibodies they produce. So from that,
Well,
we had a whole
let me
bunch.
let me break it
Oh
down
yeah,
just a little bit because my understanding, in back to 1994 is when they just went on a Western blot. It was 5 out of 10 bands,
yeah,
um, and or 2 out of 3 IgM bands. But over time, there was a lot of science that was developing at the same time because I was always fascinated when they were discussing something called outer surface proteins,
mm-hmm,
A and C. And
mm-hmm,
with that is that it seemed like the tick could carry the outer surface protein A, that type of a spirochete, and then it went to C as soon as it got into a warm body. So the vaccine was against the A, and that
right,
was some of the early work on the variety of antigens that are on the outside of the spirochete. And then there was another researcher that said that some of those proteins were changing all the time,
yep.
and they would change on their own or they would change with an antibody. So it looks like you were able to work with Dr. Embers to say, hey, there are already some of them that are tell you a lot about the Lyme disease, and coupled with what you've been doing. And so I think I just thought I'd pick up and break up into all these different opportunities to work on protein. So did I get that somewhat right as to how you got to this spot?
Right. So with her in non-human primates, she was able to identify five specific targets that did a better job than the existing Western blot all by themselves, some of which had already been used in testing. So I think there were three that were not in the standard Western blot. And then we actually were able to find, potentially a couple hundred different novel antigens that nobody, well, that's not true. Many of them showed up in other people's research, but nobody had tried to use them as a test. So we chose our top five and combined them with her five antigen panel to come up with a multiplexed 10 antigen panel. And when we put that all together and tested it against human samples, it turns out that it is actually very, very accurate. So some of them are novel, in other words, some of them come from the research that we were doing on the immunosignature. Five of them are Monica's in her published research study. It's a standard immunoassay, a bead-based
Mhm.
immunoassay. It's just highly multiplexed. And the thing that makes it very different is that it is linked to a algorithm. So in other words, we've been using machine learning to train train the algorithm what the difference is between healthy and the controls versus Lyme disease cases. And as a result of that, with the help of that machine learning developed algorithm, we can distinguish Lyme from non-Lyme with an accuracy of over 92%
Okay,
in
so
one
back to
test.
back
In
to a
one
question
test.
on that. When you do this, you take a blood sample, spin it,
Yep.
and you look for not antibodies that the body generates.
No, we're
You
looking for
you
antibodies, both IG
antibodies or antigens?
antibodies. So the antigens are bound to the beads. So
Okay.
this is a serological assay, a serodiagnostic test. But the difference is these are novel antigens, and we're looking for simultaneously IgM and IgG responses all in one test. So by the time we're done with that, we have 20 different data points that the algorithm is able to sort through to distinguish between Lyme disease cases and healthy controls.
So with that is that lots of times the labs are rather limited because
Mhm.
it proves that it works against the 20 samples or 50 samples that they have to work with. And those are samples of patients who are just very stereotypical, very like obvious. And then it falls apart when you go out into practice or when you go
Mhm.
out in the community. So it sounds like you've been able to work with a biorepository to say, well, I don't want to work with just that first small sample. I want to work
Yeah.
with a broader range of early and late Lyme, much broader than they have in a typical lab.
Yeah, so our sample set for our verification study is 364 samples. So, and the more samples we test, the stronger it gets. So that's why I'm very optimistic that this, that we're going to be successful with this. And I also want to point out, with our own experience was, the having to go outside of the insurance system in order to get testing done, paying out of pocket for so much of this, no re insurance reimbursement at the time. That this is something that we wanted to spare other people. So there are very interesting technologies that are, in the works already, but the reality is those are going to be hard to translate into a laboratory test that can be done in a clinical laboratory, like for example, LabCorp Quest, right? That's who's going to be running this test. So you as the doctor would, check the box in your office, draw the blood, send it to LabCorp Quest. They run this immunoassay and you get the result back in 24 hours in just one single test.
Well, accessibility makes a big difference
Yeah.
because there's as soon as the tests have to be sent to outside, to a specialty lab, to any lab, it gets complicated.
Yes.
And part of it is because it's often not covered by insurance.
Yep.
But it sounds like that really accessibility to a test was a premium for you and your
Yes.
team.
As part of our mission, the company's mission is to make this test accessible so that everybody will have access to it without having to take out a loan.
Well, I think that's part of the grassroots type of work
Mhm.
because you've been working at the, you know, in addition to being a professor, you've been working at the grassroots trying to
Yeah.
build up, something that's accessible. Dr. Embers is, and it sounds like the other members of your team are all on the same vision of making an accessible test that can be done and can be interpreted, which would be a game changer.
Right. And I don't think I mentioned this, but we're going for FDA clearance of this test. In fact, we're in the process right now. And we've already had conversations with the FDA, and they are suggesting that we take the route of de novo, which just basically means there are no tests because this is one tier, one test, right? One single test. It has an algorithm that is associated with it. There is no predicate test. So we're not relying on, we're not going to be, at the end of the day, we're not going to say we're as good as two-tier serology, right? We're not 98% as good as two-tier serology. What we have to do, literally, we're inventing the method to prove that our test is accurate versus not, right? So that is one of the challenges and one of the exciting parts, I think, of the de novo process. And then, I kind of look at it that later on down the line, LymeSeek, that's the name of the test, will be the de novo or will be the predicate test for other, test kit folks who want to try to get FDA cleared. They're going to be using our test as the standard going forward. That's a ways down the line, but
Yeah.
that's what I hope for.
Well, it used to be that a test could get approved as long as it was better than something else.
Or as good as,
So,
right?
like a lot of times, if you had a 35% success rate, 35% sensitivity, if you could at least break that barrier
Yep.
or get better. And so you kept having all of these tests that were well-meaning, but they got
Mhm.
stuck at 35, 40%. They would hire if you just pick self-selected samples that were very clear and very obvious. But as
Yep.
soon as you left the office, then the sensitivity would often drop to 35, 40% in my experience at best.
Yep.
And so that's why this one sounds like it's more of what they call translation, is that instead of a select samples, you're saying, well, how would it perform in a more of a general population
Yes.
than a self-selected? And so it sounds like it'll be more of what they call a robust lab than
Hmm.
just some kind of like something that comes out of a small lab somewhere.
Yep, that is the overall plan. Yep. So we at this point, you mentioned 35%. We did do a direct comparison of LymeSeek with both modified standard and modified two-tier serology. So, within the first 3 weeks, the everybody says, well, the lab tests are not accurate in the first 3 weeks because people don't produce antibodies. And what I would say is that they do produce antibodies, but they're not the ones that are on the standard Western blot. So we have identified antigens that show up, the antibodies show up early. And because we include IgM antibodies in the analysis that we are able to detect within the first 3 weeks at better than 94%. So, in other words, this is a kind of test that somebody shows up at your office with a bullseye rash. You could order this blood test, send it off in 24 hours. You would know whether the patient had Lyme disease or not and be able to prescribe that antibiotic without telling them, better go home and wait and watch, which was the medical advice that we got. Actually, my daughter didn't have she never got a rash. She had the tick bite, and we were told go home and wait for a month. And by that point, it was too late because the bacteria, as a microbiologist, as you know, the bacteria disseminate, go to places and tissues that are where they change everything about themselves and they disrupt the immune response, the overall immune response. And that's one of the drivers, I think, of the chronic illness that goes along with Lyme disease. So, early detection, early identification, and early treatment seem to be the best option for preventing that, from preventing long-term chronic illness. So that's why I personally am very excited about the fact that we are that accurate that early.
Yeah, I think that the Western blot that was codified in 1994
Yep.
when I was young, and they picked 5 out of 10
Mhm.
um, well-known well-known kind of an antigen type of things, and so but it was decided by committee, so that wasn't adaptable. We're
Mhm.
working with the same bands, the same criteria that was all decided in 1994. So the nice part about it is that you're setting it up to be algorithm driven, data driven, and so therefore, when I asked the question like, well, can we use it as a marker of severity? Or can we use it as a marker of someday where it shows a success? We don't have tests like that, but I would assume you must have on this your dream list beyond getting it approved. Will it have some other value than just yes or no? So I imagine you'll be looking at that at some point.
Yes, and in fact, we already have, but our focus at this point, because we're a very small company, and we're we tend I tend to roam, and I want to do all these different things, but we really need to stay focused on getting this test out. And once we do that, what I can tell you is that, well, and again, you probably already know this, the immune response to the infection changes in one person, right? The same person infected, it will change over time. And so with what we're doing is that the algorithm can pick that up and we can actually see that. So eventually it will be with the right algorithm, it would be possible, it's going to be possible to determine, later on down the line if the person has gone into a chronic phase or other things. But that, like I said, that's a ways off, at least 2 years, because that's what we anticipate it's going to take us about, well, actually about a year and a half, right? So we're hoping to have the test available as a commercial test kit by the end of 2026. So,
I can't wait. So there's always delays, but doesn't mean I can't be eagerly waiting for that data that is available.
right. Well, and just so for the record, we have a timeline and we have been on time and on budget for every step along the way so far. So I know things can happen, but so far so good. So
And what's that? Do you have a vision of what that test is going to be called? A working name?
so the name of the test that we have decided on is LymeSeek.
Okay. LymeSeek.
Yep, we're seeking Lyme.
Okay. And you have a website that discusses your work, your team, everything about it. So I'm going to put it in the comments below, but can
Okay.
you say it verbally?
Sure, it's Aces Diagnostics, all one word.com. And Aces is ACES, which is the combination of the first letter of all of our last names arranged as a word. It's the only word that you could make out of for our initials. So that's why it's Aces Diagnostics. And so it's acesdiagnostics.com.
So A doesn't stand, is that you?
Yeah, that's
Does that
me.
mean that you're number one or just the
Well, at
first
least in the
letter?
name.
Okay, that's it. Okay. Well, it's
Tammy Crawford is our CEO, and she's really the driving force behind the
yeah.
company. And she's truly a force of nature, and that's why I'm very convinced that we'll be successful in this venture.
What's her
So,
name?
Tammy Crawford,
Oh yeah, so I know Timmy. So
yeah,
yeah,
you've
one.
met
It's
her.
always nice to have different people with different strengths on the on a project. So she's a lot of the administrative, the technical, the scientific, and a lot of those things just to try
And
to
the
keep the
the
team
wind
together.
behind the sails, yes.
Yeah,
Yep.
so you certainly put together a good team. Maybe because of all that work you did for years, is that you could identify a team that each could come together with the right chemistry.
Yeah.
So is there any other items that you wanted to present? Because I just didn't want to miss something.
No, I mean, something a lot of the success of this, what we've done so far, we did like I said, we had a biorepository IRB set up where we actually invited people to donate samples. It was up here, up near Albany, right? So we did a lot of that up there. And I just want everybody to know that spent the time, and there were 450 people that came out to do that, okay? So all of those samples, the people that showed up, rolled up their sleeves, let us stick needles into their arm and take their blood. The success that we've had, we couldn't have done it without those samples. So I just hope that anybody that might hear this that would participated in that blood draw clinic back in 2017 and 2019, we did two of them. I just want them to know that letting us stick needles into their arm was really the force behind this. We couldn't have done it without that. So
Yeah, it's nice that your team has been able to work with something important, but also work with the people, with the patients, with those who have a vested interest, I'd say, and so in getting healthy and getting well and having tests. So proud of what you've all been able to accomplish so far, and all the barriers to get to that spot. So I,
well, back at you because, when 15 years ago, Lyme was it was a whole different landscape. And I like to hope that, all of us, you and I and everybody that's been, trying hard to change that landscape over the time, I'd like to think that, things are different now. They really are. There's acknowledgement that the tests aren't very good. There's acknowledgement that there is a Lyme-associated chronic illness. So the landscape is changing, and that's going to be a good thing.
I think that one last thing I'll say before we close is that there's been a lot of discussion of calling it post-treatment Lyme disease syndrome. So
Hmm.
even though there's discussion that it could very well be from an infection, the fact that it ends with a syndrome, some doctors that have been colleagues of have not really pursued the infection as thoroughly as I would like. So it's possible on my dream list that you'd have something that could take a post-treatment Lyme disease syndrome and give the doctor a little more to go on rather than it's just a syndrome. So that's on my wish list for when you get to the end of this level of what would be coming up. So
Well,
anyway,
mine too, because that was my daughter. And I actually, the National Academy of Science, Engineering, and Medicine, NASEM, they've held workshops on this. And at there was an excellent publication that came out of that where essentially they acknowledge this, and what they're calling it is Lyme infection-associated chronic illness to go along with other infection-associated chronic illnesses. So the fact that infections are being recognized as the driver of chronic illness, although, exactly the role what these microbes are playing is not 100% clear, but I've started using that term, Lyme infection-associated chronic illness or Lyme-associated chronic illness, because that's what it is. So, you know, stay tuned as to whether or not the bacteria remain present, or whether, it's funny because the research on the antibiotic studies, again, as you know, are not conclusive. I mean, the fact that they are being used to say, no, you can't use long-term antibiotics. When you look at the actual studies and the data, it's disturbing to me that it's written off as a possible treatment for that disease. So it will be a good thing, and honestly, NASM, National Academies of Sciences, was actually very interested in saying, this is real and we need to develop better therapeutics for it. So there's hope.
Yeah, so let's end with the word hope. So, Holly
Yes.
Ahern, is that I've had hope since I've met you from the very beginning. When I was young and we were looking at where to begin. So that's nice to be at a spot with ACES to work on a deliverable
Yeah.
that keeping the dreams going. So,
Yeah.
so I wanted to thank you for sharing this time with me. It was great to have it published in a paper, scientific paper, peer-reviewed.
Oh no, that was the article. So we've had two abstracts published. We are working on the
Oh,
publication.
okay.
So
So
it's in progress and hopefully, I mean, by the end of September, sometime in September, we hope to have it submitted. So stay tuned. I'll let you know when it gets published.
I'm eagerly waiting for that to
Yeah,
because
yeah,
it's already working with your peers through the traditional mechanism of abstracts.
yep,
I think there was a long discussion in Medscape also that inspired me to think and
yes,
reflect
yep.
on where you're at and where you're going. So
Yeah, that Medscape article, the writer, she did an excellent job in trying to explain the issues with that. But that was, that I presented a poster at the Association for Diagnostic and Laboratory Medicine, the ADLM meeting, which is a huge meeting of
mhm.
all kinds of scientists, but it's directed at clinical laboratories stuff. And we were one of 19 abstracts that won an award, right? So that abstract was recognized. In fact, I'm going to be doing a webinar for the infection, what is it, immunology and infectious disease division of ADLM in October. So they were very, very interested, and we were very honored to get that award. So that
Yeah.
was our first foray into the world, those first steps into getting the paper out there and getting it published and having the word done. It was nice to be recognized at that very first outing.
Yeah, to get honored so quick is it
Yeah.
shows that energy. So I appreciate you spending time with us on this all important topic that we all appreciate. So anyway, appreciate it. I think we're
Yep.
going to close. Thank you everyone who's joined us for this the meeting and have a good evening.
Thanks. You too.